Our next speaker is Professor Herbie Newell, Professor of Drug Development at the University of Sunderland and Emeritus Professor of Cancer Therapeutics at Newcastle University in England. His presentation is the discovery and development of CF10, a cysteamine prodrug to improve the lives of those with cystinosis.
An update today of our work on the discovery and development of CF10, a cysteamine prodrug to improve the quality of life of those living with cystinosis. This work was originated by our friend and colleague Ros Anderson, who sadly passed away in 2018. Ros was an inspirational researcher and scientist, and she is greatly missed. As the community is well aware, cysteamine is the only effective drug for the treatment of cystinosis. It is an effective drug and works by removing excess cysteine and delays the need for kidney transplantation. However, it has a number of significant disadvantages, including bad taste and smell, damage to the gastrointestinal tract, and ulceration. Importantly, it undergoes rapid metabolic clearance, and hence large doses and frequent dosing are needed. And finally, there is metabolism to volatile noxious compounds which cause severe halitosis and unpleasant body odour. The pharmacology of cysteamine has been extensively studied and has been reviewed previously at this meeting. Following uptake into the lysosome, it forms a mixed disulfide with cysteine, and the cysteine-cysteamine mixed disulfide is then removed from the cell on the from the lysosome on the lysine transporter. In seeking to address the deficiencies of cysteamine, Professor Anderson designed CF10, a prodrug in which the cysteamine is targeted to the cell via gamma-glutamyl transpeptidase, and both the gamma-glutamate and the thiol group of cysteamine are protected as esters to reduce gastrointestinal intolerance and also to give good oral bioavailability. Critical in vitro metabolism studies have demonstrated proof of concept and have shown that CF10 is metabolized sequentially by human carboxyesterases and then gamma-GT to release cysteamine at the target tissues. In vivo studies performed in the rat compared cysteamine, as shown here, with CF10. And following the administration of cysteamine intravenously, there is rapid clearance, with the drug being undetectable by 6 hours, consistent with the need to dose 6 hourly clinically. There is uptake of cysteamine into tissues, shown here are kidney and muscle, and the levels in these tissues equate approximately to the levels found in the blood. In comparison, an equimolar dose of CF10 produces firstly substantially higher levels of cysteamine in the tissue, with tissue to plasma ratios of 10 for muscle and 50 for kidney. Importantly, much lower systemic levels of cysteamine itself, and consequently a reduced risk of metabolism to volatile thiol metabolites. These data shown here are for intravenous CF10. However, similar data were obtained using oral CF10 as well. Due to the extremely poor and variable pharmacokinetics of cysteamine itself, oral studies in the rat could not be performed. Importantly, in comparison to cysteamine, where kidney to plasma cysteamine concentrations are approximately 1 following intravenous dosing, following both intravenous and oral CF10, the kidney to plasma ratio is approximately 50, reflecting the far superior tissue delivery. Following on from these important proof of concept pharmacokinetic studies, pharmacodynamic studies have been performed, initially in vitro, and shown here is the uptake of cysteamine into proximal tubal epithelial cells treated with either CF10, the solid line, or the parent cysteamine, the dashed line. And as you will see, CF10 is as effective as cysteamine in delivering cysteamine to kidney cells. This shows the depletion of cystine in cystinotic fibroblasts, again treated with an equimolar concentration of either CF10 or cysteamine. As you will see, both drugs produce a similar depletion of cystine, although in the case of CF10, this is somewhat delayed and prolonged, as would be anticipated for a prodrug. Moving to in vivo proof of concept studies, in a cystinotic mouse model, we see that following cysteamine and CF10, there is approximately equal depletion of cystine in the kidney, and also depletion in the spleen and the liver. And finally, an in vivo study in the zebrafish model of cystinosis shows that once more CF10 and cysteamine are equally effective in this model. So with CF10, we've achieved a molecule that has minimal smell and good pharmaceutical properties, reduced gastrointestinal damage in mice. It's active in both in vitro and in vivo models of cystinosis. Importantly, there's markedly reduced plasma levels of cysteamine following the prodrug, and that's after both IV and oral dosing in comparison to IV cysteamine. There is equal or greatly improved delivery of cysteamine both to cells and to tissues in vivo. And based on the improved pharmacokinetics of CF10, we envisage there will be a need for less frequent dosing, and there will also be lack of halitosis. Given that this would be a lifelong therapy, we have so far no evidence of cellular or genotoxicity in vitro or unacceptable side effects clinically in vivo. So what will the potential benefit be for patients? A palatable treatment, reduced side effects, notably gastric irritation, halitosis, and body odor problems, improved renal cysteamine delivery and hence lower risk of kidney failure and the need for dialysis and transplantation, less pre-systemic and systemic metabolism, and thus a lower dose requirement, further reducing the risk of non-specific side effects. We also anticipate future doses being required each day, only 2 per day, improved quality of life, greater adherence, and overall an easy to take therapy. Following the design and discovery of CF10, we are currently halfway through the preclinical development as a prelude to clinical trials. Our future studies are to complete this preclinical development over the next couple of years, at the same time to undertake a run-in study in cystinosis patients to determine feasibility, logistics, and validate all the required pharmacokinetic and pharmacodynamic assays. And then from 2022 onwards, in close interaction with our cystinosis community, we'll be moving to clinical trials. I'd like to finish by thanking the CF10 project team shown here at Sunderland, at the university's hospitals in Birmingham, Sundara Pharmaceuticals, our colleagues at Fine Organics, Covance, and HGF. And finally, our co-workers, collaborators, and those who have funded this work, as shown here. Very many thanks for your attention. Just in conclusion, I would note there is one other cysteamine prodrug currently approaching clinical trials known as TTI 0102. However, there are no clinical data available on this molecule as yet.